Thank you very, very much for that very kind introduction. And it's really a wonderful pleasure to be back in Berkeley. That still feels a little bit like home to me. And it's of course also a great honor to be allowed to deliver the FOERSTER lecture here. But I must say also that I'm a little bit intimidated by the subject matter when I got this letter about the immortality of the soul. And I think I sort of have to announce right at the start that I will not talk about the soul at all. So what I will then discuss is our attempts to study the genome, the DNA of Neanderthals. And as you will know, these are the closest extinct relatives of present-day humans. They are the closest relatives that are extinct to all humans that are around on the planet today. This is the Neanderthal skeleton on the left compared to modern human skeleton. They are these sort of robust forms of humans that appear in fossil record around 300,000 or 400,000 years ago in Europe and Western Asia, where they then lived till they disappeared around 40,000 years ago or so. And what I will focus on are then comparisons between the genomes of Neanderthals and present-day humans, particularly with a view to then seeing what is unique to fully modern humans compared to our closest relatives, but also then on what DNA Neanderthals have contributed to present-day humans. So as was said in the introduction, I will then perhaps, if you like, not talk about the immortality of the soul, but a little bit about the immortality of Neanderthals, if you like, in the sense of how they live on a little bit in some people today. But before I then start, I thought I should just remind you about what you all know, that our genome, the DNA, is stored in almost all the cells in our body in the form of DNA, which is replicated every time new germ cells are formed and a new individual is formed. And this DNA is made out of about 3 billion letters in the genetic code that are then replicated very faithfully. But sometimes errors are made. So when a new germ cell is formed, sometimes the wrong base is built in the daughter molecule. So every baby that's born carries something like 50 or 100 new mutations that's neither there in the mother nor the father. So and these mutations then we can observe as sequence differences between individuals in the population. So if you compare two genomes between two people in this room, we will have something like 3 million differences between two random genomes we choose. And if you then want to reconstruct the history of a piece of DNA or the genome, you can use these differences that you will see between two humans. You can add in, for example, you can add in a chimpanzee. You will find more differences, about 10 times more differences to a chimpanzee. And you can reconstruct the history of that piece of DNA with the help of these differences you observe. And you generally depict this in the forms of these trees. Very simply here, the two human sequences go back to common ancestor quite recently. Quite much further back is a common ancestor shared with the chimp too. And if you now go and study on a worldwide scale human genetic variation, the surprising finding that actually much came out of Berkeley and Alan Wilson's lab here is that most of the genetic variation you find in the world is found in Africa, although there are of course a lot less people living in Africa today than living outside Africa. Those people outside Africa have less genetic variation. And for most of the genome it is true that the variants you find outside Africa have close relatives inside Africa. But there is then a component of the genetic variation in Africa that's found only there. And the interpretation of that is that modern humans evolved in Africa, accumulated genetic variation there, and a part of that variation, so to say, went out and colonized the rest of the world. And with some genetic tricks, you can also estimate approximately when this exodus happened, and it's less than 100,000 years ago, probably more, 60,000 years ago. So this is the recent African origin model of modern humans, much designed, as I said, sort of here by Alan Wilson and people in his lab. But there is, if you like, then a problem with this model, and that is that when modern humans then come out of Africa, they were not at all alone on the planet. There were other forms of humans also living outside Africa since almost 2 million years. Most famously then Neanderthals in Europe and other forms that are less well described in Asia. So a big debate in paleontology since many, many years was then what happened when modern humans met Neanderthals. Did one mix with each other or not? And many years ago there were people that even believed in total continuity, that Neanderthals would be the direct ancestors of Europeans today. I think no one believed that for a long time. There were many people, particularly based on genetic data, that thought there was a total replacement, 0% contribution from Neanderthals, and you can imagine anything in between here, of course. So our first chance to test this came in 1997, as you heard in the introduction, when we were able to determine the first DNA sequences from a Neanderthal specimen, and not just any Neanderthal, actually the Neanderthal from Neanderthal, so to say, that was found in 1856 and gave its name to this group of hominids. And I can also perhaps say that we're very lucky, I think, that our first Neanderthal we studied was the type specimen that gave its name to this group of hominids. Because almost invariably, what other Neanderthals we ever have studied since then, there's always some paleontologists that come to us and say, oh, it's a bit too robust, it's a little too gracile, there's something wrong with it, it's not quite a typical Neanderthal. But if this is not a Neanderthal, then it doesn't exist. So at the time, we took a sample from the humerus up here. At the time, quite a big sample, not this big. This was both for carbon dating and for the DNA. Nowadays, we take much smaller samples of this. You work under clean room conditions to avoid contaminating your samples that contain then only traces of degraded and chemically modified DNA. Much less DNA often in your sample than in, say, a skin fragment that may be like a dust particle in the air in a normal room. And we focused on a tiny part of the genome, the mitochondrial genome that's inherited only from mothers to offspring, a particularly variable part of that. Reconstructed it cumbersomely at that time with the technique of the time, amplifying short pieces, seeing those differences that are consistently there, and then estimating such a tree of relationships for this part of the genome, for the mitochondrial genome. And what we found was then that the mitochondrial genomes of all present-day humans go back to a common ancestor between 100 and 200,000 years ago, as has been found by Alan Wilson here at Berkeley. But the mitochondrial genome of this Neanderthal went much, much further back to a common ancestor shared with present-day humans, about 500,000 years ago or so. And since then, others, as well as we, have looked at many more mitochondrial genomes from Neanderthals. They all fall together outside the variation of present-day humans. So it's quite clear there is no people today that walk around with Neanderthal mitochondria in their bodies. So in this sense, it is total replacement here, 0% contribution. But of course, this is this tiny part of the genome. The vast majority of our DNA is in the nucleus, and it's inherited from the mothers as well as from fathers to the offspring. And the chance to begin to look at that came in the around 2005 or so, with new technology, high throughput DNA sequencing technology that allows you to sequence millions and even billions of DNA molecules rapidly and inexpensively. So you can then just extract all the DNA from such a fossil, sequence randomly all the DNA molecules in it, make yourself a little DNA database, and compare it to, say, the human genome, to bacterial genomes, chimpanzee genome, and so on. The first place where this worked was in southern Europe, in Croatia, Vindia Cave here, from this bone here. It's a very late Neanderthal, around 40,000 years old. And the first thing that you will notice then in these DNA fragments is that they're very, very short, 50, 60 bases long, whereas from a blood sample from me, you could easily get 10,000 base pair long fragments. You will also notice that only a tiny proportion of all the DNA in the bone comes from the Neanderthal. Our very best bones, something like 3 or 4%, all the rest are from bacteria and fungi that colonized the bone after the death of this individual 40,000 years ago. So we were very lucky then to get funding for a 5-year project trying to reconstruct a complete genome of the Neanderthal. So we worked very hard on methods to improve how we extract DNA from the bone and make it into a form that we can feed into these sequencing machines. And they also got more efficient in how many molecules they could sequence over this time. We looked through many archaeological sites and many bones and focused on 3 bones from that cave in Croatia from 3 different Neanderthal individuals. We sequenced a bit over 1 billion DNA fragments from those bones, most of them then from microorganisms, and then matched these fragments to the human genome, taking into account that some of these had chemical modifications that may give errors in the sequences. And at the time, we then had about 3 billion base pair sequenced. So we had random fragments from all over the genome. Sometimes we had 1 fragment covering a piece in the human genome for comparison, sometimes 2, sometimes even 3, but we also missed big parts. So at that time, we then had a little over half the genome. But it allowed us to get an overview and begin to ask these questions one was interested in. So the first question was then this thing, what happened when modern humans came out of Africa and met Neanderthals? Did one mix or not? And we put together a big consortium of different groups, theoretical groups that helped us study this. K.Q. Monteslakin and his group here at Berkeley, Rasmus Nielsen here at Berkeley, David Reich and his group at Harvard, and addressed this question in actually 3 different independent ways, because we knew this was a very controversial question, so we wanted to get it right. I will just present one of them, the most direct one here, saying that an expectation, if Neanderthals mixed with ancestors to Europeans, would be that Europeans should share more genetic variants today with Neanderthals than Africans today would share with Neanderthals. Because Neanderthals have never been in Africa, so there's no reason to assume they would have contributed anything to Africans. So it's this idea here, if there was a contribution from Neanderthals to Europeans, Europeans would on average be closer to Neanderthals than Africans here. If we go back here. There's no contribution, the Neanderthal is equally far from people in Africa as people in Europe. So we then sequenced not only the Neanderthal genome, but 5 people from different parts of the world to be sure we had exactly the same types and frequencies of errors in the sequences. A European, 2 Africans, a Chinese person, and 1 from Papua New Guinea, and did a very simple analysis. So just to test this first, we take 2 Africans, we just compare those 2 African genomes and find all places where they have differences. Then we take the Neanderthal and see how often does the Neanderthal match one African or the other African. Since Neanderthals have never been here, there's no reason to assume that Neanderthal would have contributed more to one African than the other, so this should be 50-50 matching. And indeed, statistically speaking, that's 50-50. It's no difference between these 2. But it was then different when we looked at a European and an African. Now we found statistically significantly more matching to the European individual than the African individual, which surprised me at the time. I was really biased towards thinking there had been no contribution. But I was even more surprised then when we compared a Chinese individual to an African individual. We again found more matching to the Chinese person, although most people would say there have never been Neanderthals in China. And even more surprising than Papua New Guinea, where for sure there have never been Neanderthals, we again see more matching to the Papuans than to Africans. So the hypothesis that came out of this was to say that if modern humans come out of Africa, they presumably pass by the Middle East. And we know there has been, so this was the question, how could this be? And the model was, we knew there had been Neanderthals in the Middle East. So these early modern humans that came out, mixed with Neanderthals, and then became the ancestors of everybody outside Africa. These modern humans could have carried with them this Neanderthal contribution, so to say, out to the rest of the world, even to areas where there hadn't been Neanderthals, to the extent that somewhere between 1% and 2% of the genomes of everyone outside Africa today come from Neanderthals. So what you can then see that there is this sort of parts of the variation in Europe, for example, that match Neanderthals. This isn't so easy to see. This is a schematic picture of variation among a couple of hundred Europeans. Any sequence difference is indicated in the genome by yellow here. On the bottom is the Neanderthal genome. And you will see that there are a few European here for this region that's almost identical to Neanderthal and quite different from everyone else. There's sort of these types of fragments that sort of make up the evidence for this. But this was not the only possible model to explain this. There was also the possibility that that could go back much further to substructural populations in Africa. If we imagine that Neanderthals, maybe 500,000 years ago or so, have an origin in Africa and leave Africa and become Neanderthals in Europe. And that this substructure survives in Africa, and that then modern humans evolve in the same corner of Africa where Neanderthals did, leave Africa and meet the Neanderthals. And these modern humans also spread around the rest of Africa and absorb this variation. We could arrive at a situation where Neanderthals are closer to Europeans today than Africans are. This is a more complicated explanation, but it's a possible explanation. But the difference here is then the similarities between Neanderthals and non-Africans would be quite old, several 100,000 years old. Here it would be quite recent, less than 100,000. So a very big question early on became, when did this admixture happen? And David Reich and Nick Patterson and their postdocs, Sriram Sankararama, came up with a way to address this. And that is to say that if Neanderthals and modern humans mixed, the first generation, the hybrid, so to say, will of course have one chromosome that comes from Neanderthals and one from modern humans. If these individuals then continue to have babies with the modern humans, there will be recombination in each generation, so that information is crossed over between these two chromosomes. So there will then be mosaic chromosomes with part Neanderthal, part modern human parts here. And as the generations go on, there's more and more crossing over happen. So these pieces get smaller and smaller with time. So you can actually look at the size distribution of Neanderthal fragments and estimate how many generations back were they introduced into the modern human gene pool. So look at the sizes of these things. So you have to make a number of assumptions here about generation time, recombination landscapes, and so on. And making those assumptions, they came up with a date somewhere between 40 and 90,000 years ago. So quite recently. Only compatible really with this recent admixture to modern humans when they come out of Africa. But I'm a sort of really practical person. These are, of course, theoretical considerations that rely on a number of assumptions. And maybe some of those assumptions are wrong. So I really like to go back in time and try to find actual evidence for when this happened. And that is beginning to be possible now. Because there was a bone found now just 2 years ago in a river in western Siberia here, at a place called Ust-Ishin, at this Irtysh river. And there was a bone washed up on the shore there, a femur like this, that looks very much like a modern human femur. And we were very lucky to get this femur to our lab in Leipzig, and we were shocked when we radiocarbon dated it, because it turned out to be 45,000 years old. So it's actually older than any other directly dated modern humans outside the Middle East and Africa. So this is a very early modern human, 45,000 years old. So falls actually in this range, 40 to 90,000 years ago, when this mixture with Neanderthals could have happened. And certainly this individual lived at a time when there was also Neanderthals around. So now we can, and this is a sort of unpublished part of what I'm telling you, look for the first time in an early modern human that lived at this time and say, have this individual met and bred with Neanderthals or not? So if we first look here on just one chromosome, a number of European and Asian people today, Neanderthal fragments are indicated by red, when there are homozygotes, they are green here. You can actually see a difference here between Europeans and Asians. If you squint a bit, you can almost see that the Asians have slightly more Neanderthal fragments. And that's actually true. There's sort of evidence from the Svatne's lab here that Asians and others also have shown that Asians seem to have an additional Neanderthal component that you don't see in Europe. But the big question for us now is this Austrasian individual here, 45,000 years, does that have any Neanderthal contribution or not? And the answer to that is yes, it does. And actually a lot, if you like. Overall, not more than this individual, but it's distributed in much bigger chunks, as you would expect, right? When you go back in time, there should not have been so much recombination having happened. It is this thing again. So we can go back to this scheme, sorry, and look at the length, if you like, here, in genetic length on the chromosomes of Neanderthal fragments, and how well the Neanderthal variants correlate over length of the chromosomes. If you look at these are present-day humans here that arrive at this 40 to 90,000. This is this 45,000-year-old human now that allows us to estimate that somewhere in the order of 300 or 400 generations before this individual lived, Neanderthals contributed here. So that would then allow us to estimate this to somewhere 50 to 60,000 years ago. So we're sort of narrowing in on when this happened. But very clearly it's this model that is right then. There's genes from Neanderthals to modern humans after what comes out of Africa. So there are a number of questions you all often get about this. Some people sort of ask, is this a lot of Neanderthal ancestry I have, or a little, when we say 1 or 2% of our genome? And one way to think about that in very simple terms is perhaps to think about your own family tree. If this is you of course share 50% of your DNA with your mom and your dad, 25% with your great-grandparents, 12% approximately with your great-grandparents, with your great-great-grandparents, 6%, 3%. And if we now go back 6 generations, you then share on average 1.5% or so. So in some sense, in a quantitative sense, it is as if one of your ancestors back here was a Neanderthal. It's of course much further back and it's distributed differently in the genome, but it might give you a feeling for sort of how big a contribution is this. So something else that people often ask us is, well, were the Neanderthals the same species as us or not? Sort of, should we call them a separate species name, Homo neanderthalensis, or a subspecies? And I always sort of dodge that question and say, you can even cite Darwin about this, and saying to discuss if two things are right equal species or varieties before we have a definition of these terms is to vainly beat the air. And we still don't have a good definition of this, so I sort of don't answer that question. So another question then is sort of how much of the Neanderthal genome remains today? Because if your roots are outside Africa, you have 1 or 2% of the genome from them, but we carry different pieces of it. So if we walk across many people, how much of the Neanderthal genome can we reconstruct from people today? And there were 2 papers that came out in January, one from Josh Akers' lab in Seattle and one from David Rice's lab that we were involved in, sort of doing that in the 1,000 genomes data. So you can sort of go across many people on the chromosome here and see which fragments come from Neanderthals and puzzle together pieces of the Neanderthal genome from people living today. And it's very, very clear that you can get together at least 20% of the genome, and probably twice that, I would think, because we have big problems recognizing short little pieces, so we probably miss several pieces. So perhaps approaching half their genome or something like that is still going around on the planet today. That's the immortality part of the talk. So I also can never refrain from saying that there are a lot of people in the general public that are fascinated by this and write to us. Many people write to us and self-identify as Neanderthals and volunteer to give examples to us. And I have started very early on to notice a pattern in this correspondence. And that is mainly men who write to us and say they're Neanderthals. And very few women who say that. So I sort of presented this as my research to my group, I counted emails. But people are generally critical, particularly when I have ideas in the group. So they said this is just ascertainment. Men are more interested in molecular genetics, and they write to you, and women are less interested. But that's actually not true. If I go back and see, there are actually quite a few women who write to us and say they are married to Neanderthals. And so far there is not a single man who has claimed he's married to Neanderthals. So that's, of course, quite fascinating for a geneticist to have. But something else that we're also interested in addition to counting emails, is then other extinct forms of humans. And particularly we are lucky to work with people in Russia, particularly at this site in southern Siberia, the Nisova Cave, this beautiful place close to the border of Mongolia and China, where Anatoly Derevianko and Mikhail Shokov excavates in several years. And they found this tiny little bone in 2008 in this gallery in the cave here, that they were actually very skilled realizing that that might be a human bone. So we got this fragment of this pinky of a child, the last phalanx of the pinky of a child, and analyzed its DNA. And it was quite unique in actually having 70% endogenous DNA, so very little bacterial growth in this one. And we applied some new technology we've developed that actually starts out by separating the two strands of the DNA molecule and sequencing, putting them into these libraries for sequencing individually. So each double-stranded molecule have two chances to end up in the library, one for each strand. And with this method, we were then able to sequence a very good genome from this individual. So for the part of the genome, about 2/3 of the genome to which you can map these short fragments, we've sequenced it many, many times over, about 30 times over, so we've seen every position several times. And we were extremely surprised then to find that this was not closely related to Neanderthals and even further from present-day humans. It shared a common ancestry here, far back with Neanderthals, but Neanderthals then have a long independent history. It was clearly some other form of human related to Neanderthals. And we, after much sort of discussion about this, we ended up calling them Denisovans after this cave site where they were then first seen. Just as Neanderthals are called Neanderthals after Neanderthal that they were found. And there are a number of interesting things you can now do when you start having really high quality genomes from humans that lived a long time ago, as high quality as you will sequence from a patient today. You can, for example, start seeing, as you would expect, that this individual died several tens of thousands of years back. So compared to people today, there are sort of mutations that have not happened here. It sort of lacks mutations. So if you go back to the common ancestors with the chimp, a bit over 1% less substitutions than you would expect. You see sort of evolution in action, if you like here. So if you now just make the assumption this is 6.5 million years ago, we can even date the bone here to somewhere 60 to 80,000 years. This is with big caveats. I don't have much faith in this number at all. You also see variation here, showing we have problems with sequence accuracy in the modern humans, for example. But it anyway gives an indication of what will be possible in the future. So this bone is far too small to be able to do a carbon date. But when we can do a good genome from it, we can actually in the future even date it, and all with genetic means. You can ask for these Denisovans, have they contributed to people today? And indeed they have. Surprisingly, although the bones are found here in Siberia, we find very little contribution in mainland Asia. There's now new evidence that there's a little bit, but the major contribution is out in the Pacific. Papua New Guinea, Aboriginal Australians, up to 5% even Denisovan DNA. So if we just summarize what we believe from studying genomes about the origin of Neanderthals and Denisovans first, we believe there's some origin in Africa. They come out of Africa and in Western Eurasia evolved into what we call Neanderthals, and in Eastern Eurasia to what we call Denisovans. This is not at all to say they were this widely distributed at any one time. Also not that there are not other forms of hominins here. We have the hoplites in Indonesia, for example, that we know about. And we don't know where the borders between these have been, but we do know that in this part in Siberia there have been Neanderthals as well as Denisovans at some time in history. Then modern humans evolved in Africa, start spreading out of Africa seriously 50, 60,000 years ago, mixed with Neanderthals in the Middle East, continue to spread out and mix again at least once, it seems, perhaps somewhere in Central Asia or so, with the ancestors of present-day Asians, and mix in Southeast Asia, somewhere with Denisovans, and continue out in the Pacific. And these old forms then become extinct, but live on, if you like, a little bit in that 1 or 2% in all non-Africans today, and an additional 5% or so in the Pacific. Now, we have studied 2 genomes of 2 extinct forms of humans. I wouldn't be at all surprised if one find additional admixture events, for example, in China. And I would also not be surprised if it was the case that in Africa, when modern humans spread there was also admixture. There's some indication of that in present-day variation in Africa, but it's much harder to really demonstrate when you don't have these old genomes. But so in this sense, then, we clearly disproven the total replacement. We have something like 7.5% maximally or so far. But the big picture is still one of replacement, right? So to not lose track of that, to sort of like to suggest a sort of model called feature replacement or so for how modern humans spread with some gene flow then from these other forms. So I think it's sort of interesting. This is a copy of this little bone, that from such a tiny little bone we can actually get a lot of information about the population history of the population from this individual came. But very frustratingly, we don't know how this individual looked. We don't have no other parts of the skeleton except 3 teeth, actually. We don't know what stone tools they made. We don't know anything about them except the genome and much of the population history. And I think this is an indication of how it will be in archaeology a lot in the future, that when you can retrieve DNA, also from small undiagnostic pieces like this, you will be able to get a lot of information. So this cave, the Denisova cave, had also yielded more interesting fragments. This is a toe bone that was found in 2010, deeper down. This turned out to be a Neanderthal bone. So we now also have a good Neanderthal genome that we published in January this year, where we have a very high quality Neanderthal genome. So you can look at different aspects of this also. You can look, for example, on heterozygosity, so how much variation there is between the 2 versions of the genome that this individual inherited from the father and the mother. You will see present-day Africans here have more variation than non-Africans, and here is a Neanderthal and a Denisovan having even less. And not only that, when one looks at the chromosomes of this Neanderthal individual, you find something amazing. You find long, long segments, 19 million base pairs of this chromosome, where the two chromosomes are identical to each other. And that, of course, indicates that the parents of this individual were closely related. So, Montes-Latkin and his group here at Berkeley have modeled what the relationships must have been between the parents of this individual. And it has to have been one of these four scenarios here. The parents were half-siblings, or grandfather, granddaughter, or some of these things, double first cousins. I can't even reconstruct what it is. But they were clearly closely related. So I think it will be very interesting in the future when we get good genomes also from other Neanderthal sites to see if this is a general pattern at the time, or this was something just happening in this cave at this one time. So what we then have are these good genomes from Siberia. We have a bit of a genome from Caucasus and this one from Croatia. So we can now start to look at gene flow not only from these groups into modern humans, but also between them. So what we find then is what we already know, this gene flow into non-Africans from Neanderthals, from Denisovans into people in the Pacific. We find a little bit of gene flow from Denisovans into mainland Asians. So people in China, for example, also have a little bit of Denisovan DNA. We find gene flow a few percent from Neanderthals into Denisovans. And quite interesting, an old component in the Denisovan genome not seen in the Neanderthals. There is some gene flows from someone else here of a few percent into Denisovans. Something that split 1 million years or more from the human lineage. So it's very tempting to say that this unknown contributor here is Homo erectus we saw in Asia contributing. So the conclusion is human groups have always mixed, at least a little bit. And in the end then I wanted to bring up 4 things, the sort of consequences of this gene flow. So the question is then, we have this Neanderthal component in the genome outside Africa today. Does that have any functional consequences? And there are some hints, beginning to be some hints in the last few years that that may be the case. So there was this paper in January which looked across the genome in Europeans and in Asians, finding that there were some parts of the genome that actually where there are contributions from Neanderthals that have reached high frequency, 60-70%. 60-70% of Europeans have that. Sometimes it's shared between Asians and Europeans, sometimes it's unique to one group or another. So of course, one interesting thing is to look in these segments and see what genes are particularly located there. If you look for groups of genes, the only thing that stand out are actually structural proteins, keratins, in skin and hair. So it may well be that something in the structure or of the skin or the hair in Europeans and Asians actually is a contribution from Neanderthals. One will then find out in the future what that is. Something else that Peter Parham's group found early on, 2011 already, is that transplantation antigen genes, some variants of genes involved in regulating the immune system, come from Neanderthals and also from Denisovans, and have sometimes reached high frequency in certain regions of Europe. These are then proteins that present peptides from bacteria and viruses to the immune system. So they're important in regulating how well we respond to infections. So it's quite tempting then to say that this is something to do with these Africans coming out, meeting people that have lived for hundreds of thousands of years in an environment in Asia and Europe, picking up immune regulatory genes from them, and when they are advantageous, they rise to high frequency. There's a group in Shanghai who have shown that if you look at genes involved in lipid catabolism, so how you degrade lipids, they have a big tendency to come in Europe from Neanderthals that you don't see in Asia and of course not in Africa. We don't know at all what this means metabolically, But something may be going on. There was another study in January this year that identified a risk variant of type 2 diabetes, so the type of diabetes you get at old age. And this risk gene encodes a lipid transporter that sits in the membrane and have 4 amino acid differences to the protective allele. And quite strikingly, this risk allele is high frequency in Asia and also in Native Americans, very little in Europe and not in Africa. And if you look at the risk alleles here in red, the protective alleles in blue, you find the Neanderthal variant in the middle here. So this is clearly a variant that had come over to modern humans from Neanderthals, have risen to 25–30% frequency in Asia and Native Americans. Today it's associated with a risk for type 2 diabetes, but probably in a situation where you have periodic starvation, for example, such variants may be actually advantageous. This may be that this is some kind of Neanderthal adaptation to starvation. You can, of course, also ask, have you a contribution from Denisovans too? And quite fascinatingly, it has come out quite recently that it was already known that people on the high plateau in Tibet are adapted to living at low oxygen tensions. And that one of the major genes involved in that is its transcription factor, EPAS1. And Rasmus Nielsen's group here in Berkeley have now shown that this variant of EPAS1 that exists in 70–80% of Tibetans come from Denisovans. It's identical to Denisovans here. So this variant seems to have come over from Denisovans into Tibetans. So it's quite fascinating that even life on the high plateau in Tibet might actually not have been possible or so easily possible without this genetic contribution from Denisovans. So this sort of fits into sort of a pattern of adaptive introgression, if you like, where these earlier forms of humans have lived for a long time in these other environments, and these newcomers from Africa may have picked up variants that were then positively selected and rose to high frequency. So you can, of course, also ask what is not coming from Neanderthals. So you can look across the genomes and look for things where you statistically see a lack of contribution from Neanderthals, or you would expect to see it there. And David Rice's group had identified a number of these regions, and when we looked where genes in these regions are expressed in the body and on tissues, the only thing that stands out are testicles, so the German, male germline. So it's very tempting then to speculate that the hybrids, the Neanderthal modern human hybrids, may have had some problem with male fertility. Because that's, of course, a common pattern when closely related populations or species have hybrids. It's often the males who have problems with fertility. If you think about horses and donkeys, for example, the male mules are infertile, the females can have offspring. And finally then, the thing that interests me very much is what can we say about uniqueness of modern humans now when we compare ourselves to our closest extinct relative? So what has changed here in modern humans in the short time since we separated from Neanderthals, maybe 300,000 to 400,000 years ago? And that may be an interesting set of things, because I think we all agree that our technology, for example, with modern humans has changed very rapidly. Neanderthals lived for 300,000–400,000 years. Their technology was not that different in the beginning of that time, and in the end, modern humans have existed for perhaps 100,000 years, and I think our technology today, we agree, is quite different from what it was back then. Art that really depicts things that we recognize as art comes from modern humans, spreading across the world, becoming extremely numerous, spreading across water over long distances, come with modern humans, and so on. So it's very interesting now to make this catalog of all the changes in the human genome that have come since that time. So now focusing on these things that exist in all humans, no matter where we live on the planet, But with the Neanderthals, look like the apes. And that's not a very long list of things. These things that then happened here became 100%. It's in total just a little over 30,000 single nucleotide changes, some insertions and deletions and so on. So it's very sort of fascinating that you can look through this list in an afternoon in a computer actually. You can of course, for most part of it, not make sense of it. We don't know what these things cause. But among them, I believe there will be some important things hiding. So for example, if you look at the amino acid changes, there are only 96 amino acid changes in this category that affect 87 proteins. And this is a list of these proteins here. So we're of course biased to think that something with cognition is particularly interesting. So the developing brain was something we were very interested in looking at together with the people at the Allen Brain Institute. And originally I was very excited to see that 88% of these proteins were expressed in the developing cerebral cortex. But we then did some controls, for example, finding genes with no amino acid change, but a silent change ascertained in exactly the same way. And 100% of them are expressed in the cerebral cortex. This just goes to say that almost everything is expressed in the developing brain. But with appropriate controls, the only one that sort of seems to stand out are things that are expressed in the ventricular zone of the developing cortex and that have some kind of gradients, temporal gradients. So in the layer of the developing cerebral cortex where cells divide, stem cells divide and form neurons, we see significantly more of these genes expressed. And this relies on very few genes now. It's actually only 6 genes that are responsible for this signal. All in all, it was only 87 genes, right? And strikingly, 3 of these genes, 2 of them are in the kinetochores and 1 in the spindle. So this is sort of where the spindle attaches to the chromosomes and pulls the chromosomes apart at cell division, which surprised me very much. I thought cell division would be so conserved and not have changed with modern humans. But of course, there are evidence that in the developing cortex here, how the stem cells divide, the plane of division, for example, determines the number of neurons and types of neurons that are formed. So it may be, this is all speculation of course, but that these 3 genes with their changes is something one should look particularly careful at in the future. So we can also now begin to look, taking variation in Neanderthals into account. So we have then genomes from 3 Neanderthals here and the Denisovans, and we can begin to look for groups of genes that have changed, particularly one group of hominids and the other. And if we look in Neanderthals here, there's only one such category of genes that seem to stand out, and these are genes involved in hyperlordosis, so how much your spine is curved. And here are more things back here in sort of morphology and in metabolism. But this is quite interesting because this is something we can look at in the fossil record, the curvature of the spine. And indeed, Neanderthals differ from modern humans in having less of a curvature in the spine. So since this sort of makes some sense, we were quite interested to see what categories come here in modern humans. And there come only 2 categories there. One is sort of behavior and one is pigmentation. And again, it relies on very few genes, just 5 genes. 2 of them have to do with pigmentation, but they are not actually totally fixed. When we now have gone on and looked in Africa much more carefully, a few % of people in Africa have an ancestral variant. So this is not so interesting. It probably has to do with differences in pigmentation. But these genes involved in behavior seem to be really fixed in humans. You can of course look at what they are involved in your processes, what diseases they are involved, and you then arrive at the last question here. How will we in the future study these things that may have to do with human-specific traits? And I've sort of gone around for 10 years now making jokes and talk and saying what we want to do is of course to put Neanderthal alleles into transgenic humans. and human alleles into transgenic chimps and study their phenotypes. And that we have problems with ethics committees and things like that. So we'll never be able to do that. But this has sort of been a joke, but it's almost less of a joke now, because there are people who sort of suggest that we should now use stem cell technology and high throughput mutagenesis to actually clone Neanderthals. Research, for example, at Harvard. So I think we have exactly the same issues there. For many, many reasons, technical and ethical, we will never do that. But this is not just a joke. This is, of course, what one would have done in Drosophila or in some other small animals. So what can we hope to do in the future? I think one thing we will be able to do is find back mutations in humans. As we said in the very beginning, every baby that's born has 50 or 100 new mutations. There are 7 billion people on the planet. The genome is just 3 billion base pairs. So every mutation compatible with human life exists out there. We just have to find them and study them. And that, I think, will be possible in the future when we all have our genome sequence when we go to the doctor. But that's a little bit away. I think we can introduce these things into stem cells and study cells in vitro. And I think we can also introduce them into mice and sometimes make sense of it. And I see that that's going over time. So I will sort of not really show you then one such gene which has changes that happen back here and seems to have to do with language and articulation. Those mutations we have sort of as a model put into a transgenic mouse. And you then have a mouse that makes its, excuse me, I can keep going, but that's you say that. I don't know what the other people say. So these mutations then put into the mouse. So the mouse now makes a human version of this protein that seems to have something to do in humans with language and speech. And you can then study that mouse, the brain of it, electrophysiologically, for example, and find that you have certain electrical features of the neurons have changed. And you can also see that these neurons make longer connections in certain parts of the brain. And those parts of the brain are involved in motor learning. So these are these cortico-basal ganglion circuits that seems to be changed. So you can then develop a theory about what this caused. And there's some new data now that's not published by Christiane Schreiber together with Anne Graybill at MIT, where they study motor learning in these humanized mice that have this humanized protein down. So these are experiments where the mouse have to learn that it should go towards a certain light signal to get some reward here. And if it should always go to the left, after a while you can take away the visual cue and the mouse will automatically go to the left. It has sort of automated this. And if you look at how quickly you come to this automation of the thing, it's quite a difference between the humanized mice to the wild-type litter mates. They learn in 7 to 8 days to do this, what takes 11 and 12 days in the wild-type litter mates bred by the same mother, etc. So this is sort of the switch from this sort of declarative to the procedural, it's sort of automation of motor learning, if you like. So if you think about learning to bike when you're a kid, you first think about what you do and you're very bad at it, and after a while you sort of automate the thing, and then you get very good at bicycling. And there's this switch here to sort of automating motor movements. And you can, of course, sort of speculate and say that that's exactly what we do when we learn to speak as children. We learn to automate very complex coordination of motor movements in our vocal cords, our lips, our tongues, to produce articulate speech, something that no other ape can do. So there's an hypothesis that these changes alters these cortico-basal circuits to allow for faster automation of motor movements and perhaps some aspects of language and speech. So I think this is very encouraging that one can perhaps sometimes develop models of humanizing aspects of the brain in a mouse. So we're trying to do that with these genes, for example, for modern human changes now, and for a number of other genes that may be of interest. So to end then, I'm going to say that if you're interested then in modern human origins, I hope I sort of convince you that it's quite useful to have the genome or a very closest relative, because they can focus on what's really unique to modern humans relative to them. In the future, when we have more Neanderthals, we'll also be able to study what's unique to the Neanderthals. But that will not be enough. We will have these changes and we will have to go after them functionally. And the way to do that will be to modify human and ape cells in tissue culture, I think, but also then to modify and humanize mice. And with that, I should just say that many, many people are involved in this, more people than I can mention. I'll pick up one person, Matthias Meyer, who developed the technology to make these ultra-sensitive libraries that allowed us to get these high-quality genomes from Denisovans and Neanderthals. Many, many people have been involved in analyzing the data. I mentioned them in the talk several times, Dr. Slatkin and his group here, and David Wright and his group here, Janet Kelsu, who coordinates all the bioinformatics in Leipzig, and Kai Prüfer, who particularly then worked on the Neanderthal genome. And with that, I thank you for your attention.